research paper that I read, the researchers used an assay called 293 STF cell-based reporter assay. So today I'm going to give the necessary background information, explain what the researchers were doing, what this assay is, and how and why it was used in this experiment. So what were the researchers hoping to fix? The researchers were looking to find a way to improve the signaling pathway to fight limbal stem cell deficiency. This is an eye disease that causes pain, decreases vision, and can cause irritation, among other symptoms. Having this disorder means that the tissue surrounding the cornea is not producing the stem cells it needs to replace old ones. This leaves the cornea unprotected, which causes the decrease in vision and pain. If the cornea is exposed for too long, it can lead to blindness. So they wanted to create a mimic that helps stop this disease. As you can see in the slides, a disordered limbus means that the tissue is disappearing from around the cornea, leaving it exposed. Stem cells are needed to replace the tissue, and when they are not available, the tissue will degrade. Ultimately, the goal of this research was to create a drug that would help the pathway perform better, because this pathway is necessary to help stem cells grow and survive. As you can see, more signaling will lead to more limbal stem cells, which will replace the tissue surrounding the cornea. So what is the assay? Now that we've determined what they were doing, let's explore it. It is used to measure the activity of the pathway by expressing firefly luminescence. This means that when you add the stem cells to a test tube and then you add the firefly cells, the stem cells will glow if there is activity. When there is more activity, the cells glow brighter. If there is no activity, the test tube will not glow, kind of like the images on this slide. To help illustrate this, the yellow test tube shows what it looks like when there is activity and the tube is glowing. The clear tube represents when there is no activity, so the tube does not glow. Because their goal was to create a drug that would enhance the pathway, they wanted to see how drug 1, the green cell that we see on the slide, alone would impact the pathway. They found that it prevented the pathway because the test tubes were not glowing, meaning that there was no pathway activity. After verifying that the green cell alone would prevent the pathway, they combined it with drug 2, the orange cell, to see how that would impact the pathway. The 293 STF assay caused the test tube to glow, which told the researchers that the combination did not prevent the pathway. They also saw that when they added higher concentrations of the combination, that they glowed brighter, meaning that it helped the pathway produce more stem cells. As you can see, drug 1 had no activity, therefore the firefly cells did not glow in the test tube. Because drug 2 caused lots of activity, the test tube was glowing, and the more of drug 2 that you added, the brighter the test tube was glowing. To conclude, limbal stem cell deficiency is an eye condition that can lead to blindness. Researchers want to find a way to increase stem cell production in the eye. They used an assay to test the effectiveness of the drug they created. The assay was adding firefly cells to the drug to see if they help or hinder the pathway. The result was if the tube glows, the drug helped, and if it doesn't glow, it hindered the pathway. They found that the drug 1 did not help, and that drug 2 did, which ended up having the firefly luminescence showing in drug 2 without the firefly luminescence in drug 1. Thank you, and here are my references.